Now is we're going to take a look at how these actual action potentials and how this electrical changes, since we've talked about kind of resting potential and some of the changes that can happen with that with muscle. We're going to talk a little bit about how nerves can actually have these different types of movements, how we can actually take this information and move it through a cell, for example. So what we're going to do is we're going to start with a stimulus coming into a dendrite. We're going to have some type of neurotransmitter being released, and it's going to cause sodium channels to open up. So some of these ligand-gated channels opening up on this one. And so what we can kind of start asking is, would that lead to depolarization? What we're going to see is, because of the layout of those different channel types, what we have on the dendrites, what we have in the cell body are those ligand-gated channels. So with those ligand-gated ones, there isn't a threshold value. This is going to open up, and you're going to have this diffusion of these ions in as long as the channel is open. So the dendrites, the cell body, tend to not have these voltage-gated channels, but they're found a bunch at the axon hillock and down the axon. So what we're going to get here is we're just going to get this local graded depolarization, this local change in that area. So like at the bottom here it says, well, if that's the case, what does this depolarization do? What we're going to see is a graded potential. I always use the analogy of kind of like a house heating system in the winter. If you get it cold enough in your house, it's going to cause your thermostat to kick on. So if you open your door, you're going to get a wave of cold air that's going to go in. That wave of cold air is going to spread out within the house, overall drop that house's temperature some fraction of a degree. If you open up the door a bunch, it's going to get cold enough overall within the house, so those little cold particles are going to move out throughout the house enough to drop the temperature to kick on your furnace. A graded potential is acting a lot the same way. We get this local change, so these ions are going to flood in there. And again, they're going to diffuse out. So overall, they're going to reduce the electrical potential across that membrane some minor amount. It's going to be weaker the further it gets from where it's going in there, just like if you're standing right next to the door, the wave of cold air is going to be much colder than if you're on the other end of the house. But overall, that cold is going to spread out, just like this change in membrane potential, this change in this electrical potential of the cell is going to spread out. As that sodium ions drift, they're going to get pumped back out, and as we get further and further away, it's going to become weaker and weaker. That's what a graded potential is. It's this wave of ions going in and slowly spreading out within the cell. If it's strong enough, what we're going to see is sooner or later it can work down to the cell body, and at that axon hillock, we actually have voltage-gated channels. So if this graded potential is strong enough and lasts long enough, the change can be big enough that you could actually trigger those voltage-gated channels at the axon hillock. Once you do that, you're actually going to get an action potential happening. It's going to travel from the axon hillock down the axon to the end, cause neurotransmitter release, and that is how it's been processed. So again, with these graded potentials, they can be any size. And again, it has to be, that graded potential has to be big enough that it spreads down to the axon hillock where you have the voltage-gated channels. So if we go back to the house analogy, you have to get a wave of cold air large enough that it's going to spread out and get to the thermostat where you have that threshold value, and then it has to be strong enough to get you over that threshold value to cause this action potential to take place. Once you get this change at the axon hillock that's above threshold, it's going to initiate an action potential, and then it's a lot like how we talked about with the muscles. Those voltage-gated trip the ones next to them, trip the ones next to them, and it's going to travel down that axon to the end and cause neurotransmitter release. If it doesn't if it doesn't reach threshold, nothing is going to happen. And if we were looking at the membrane, you're going to see something like this. You're going to see this. You're going to see the depolarization happen. Repolarization, you see it kind of goes below resting level and then comes back up. So this is this sequencing of these different sodium and potassium channels. We have sodium channels opening, more sodium channels opening through here. They close, potassium opens up, stays open a little bit longer. We go back beneath, they close. Sodium potassium pumps get us back to normal. So this is going to be that kind of activation gates, inactivation gates, and the kind of the cycling through those. So sodium channels open first, they get inactivated, then the potassium channels open to repolarize the cell. That is that phase where it's dropping back down, and we get this undershoot or hyperpolarization. And again, these action potentials, once we get there, these are again, a temporary reversal of that. Those positive ions are going to flood into the relatively negative area, and then we're going to put that back to resting potential. Because these are we have voltage gated channels all along the axon, these are self-propagating. Once you trip the first one, it's like tipping over those dominoes. And again, the other thing we're going to see, if no threshold is reached, we don't get an action potential. If it's reached, we get an action potential. So this isn't all or none. Either you reach threshold, you send an action potential, or you don't reach threshold and you don't. There is no, we made it halfway there, so we send a half strength one, or we made it 2 times over the threshold as a double strength one. It's all or none. We either send the same message, that same action potential, or we don't send one. There is no amplitude to these action potentials. They are always going to be the same strength. And again, you can kind of see this little, the voltmeter, if you're looking at this, we start getting some local depolarization. It reaches threshold. At this point, those voltage gate channels are going to open. We get the sodium gates opening. It begins to enter the cell. We start getting some potassium channels open. More sodium gates open. As it gets to the top here, the sodium gates close. Potassium tends to stay open longer. We get this return to resting level. They stay open a little bit past that. We then pump it back out and we get back to resting potential. And at that point, everything is reset for another one to happen. And again, this is the cycling of these gates. These sodium channels have those activation gates and the inactivation gates. At resting potential with nothing going on here, we have the activation gate closed, the inactivation gate is open. Once we reach a threshold level, it's going to open up those activation gates and you're going to start getting sodium ions traveling through. They will continue to travel through. At some point, that inactivation gate, after a certain period of time, the inactivation gate is going to close, stop that from happening. What's then going to happen is the activation gate is going to reclose and reset. That inactivation gate is going to open up. And now we are ready for at least the sodium stuff to happen again here. During this period of time, the potassium is leaving the cell, which is putting these, at least the membrane potential back to normal. So again, 2 sensitive gates here at resting. One gate is closed, the other one's open. When it becomes depolarized, the other gate opens up. After a short period of time, that activation gate the inactivation gate will shut. So again, you see here at rest, the activation gate is closed, inactivation gate is open. We reach threshold, sodium can flood in. We then have, when it's time, after 5 milliseconds, we're going to get that inactivation gate closing to stop that, and then again it's going to reset for the next type of action potential that could be going through. The potassium channels, these just have a single gate. They have an activation gate. It's either open or it's closed. And again, we get a depolarization. This one doesn't respond quite as quickly, so you see the sodium gate is open for a little while before the potassium gate starts opening up, and then that sodium gate's going to close before the potassium gate closes, which is why we get that repolarization happening. But again, it's just a single gate. The gate is closed, potassium can't leave. We have some depolarization happening. This activation gate on this one hasn't opened yet. Finally, after a certain period of time, that gate opens up, and then again, it's going to stay open a little bit longer than the sodium gates to give us that chance to repolarize and get that undershoot, that hyperpolarization. And this is again showing the kind of cycling of these ones here. So you can see this one would be the sodium gate, this one would be the potassium gate. At rest, activation gate is closed on both of these. We go to step 2 here. The sodium gate opens up. We start getting sodium flooding into the cell. At some point during that inactivation gate then closes. And now we have the potassium gate that is open, that's letting potassium ions flood back out. They continue to flood back out. This gate is then going to close. We are then at that point reset back to resting state, and we're ready to do this again. Again, having those sodium potassium pumps helping to reset these ions back to their beginning spots. What we can see is because we have a lot of these gates opening and closing, there is going to be a certain period of time during this whole depolarization, repolarization kind of effects here, that there is going to be a certain period of time where you can't stimulate this membrane again. We're going to call this period of time where you can't re-stimulate the membrane the absolute refractory period. So what this is meaning is when you're in the process of depolarizing and starting to repolarize, you can't polarize something that is still currently depolarized. That is that absolute refractory period. So during that going up and then back down until it gets back to beneath the resting potential, you're not going to have the ability to re-stimulate that membrane. That is that absolute refractory period. It doesn't matter how large the stimulus is, that is the absolute refractory period. So what is the advantage of this? The advantage of this is we start this at one end of the axon. If this was the axon right here, we started at one end. Or let's say this is the cell body here. Dendrites, all that cell body. We start at the axon hillock right here. If we trigger the first one here, and it triggers what's next to it, and for a little bit of time, this one is an absolute refractory period. What that means is the only direction this can travel is down the axon. It can't turn around and go back. Technically, if you were to be able to take some type of electrode and put it right in the middle of an axon and stimulate the voltage gated channels there, that depolarization would travel in both directions out the axon. But because we started at the one end, this means that those action potentials will only travel one way, because as this is moving, the membrane behind it is in absolute refractory period, so that action potential cannot double back. It can't turn around until we get back to a normal level, and by that time that action potential has traveled past that area. So the other thing we have to worry about is that relative refractory period. So there is this period of time where we are back to resting potential, but it's actually further underneath that, so that undershoot. So we saw in those little things that it went up and then it went down beneath the resting potential before it worked its way back up. Could you actually stimulate a nerve impulse during that relative refractory period? The answer to this is actually yes. Once it's been repolarized, you can actually bring about another action potential. But if it's in that undershoot, what that means is, let's say resting potential is normally negative 70 and you get it down to negative 80 and you need to get it up to negative 50 to reach threshold value. If you're in that undershoot and you're at negative 80, that means you need a bigger stimulus. You need 30 millivolts instead of 20 to reach the actual threshold value. Because of that, that means you need a bigger stimulus to send these action potentials in a faster fashion. So the analogy I use on this one here is, imagine you have a toilet. When you push the handle, there's a certain threshold to this handle. If you get that handle, if you push it too late, it's not going to actually flush. You have to get the threshold of that handle. Once you actually get that, unless something messes with the toilet is going to go through a normal flush. It doesn't matter how hard you flush the handle. Once you push the handle hard enough to get it to flush, the toilet flushes the same way every time. That is that all or nothing response. And when the toilet is in the process of flushing, no matter how much you hit the handle, you can't make it flush again when it's in the process of flushing. So that is that absolute refractory period in a toilet. While it's in the process of swirling the bowl there, you can't get it to flush a second time. Once it flushes and that tank is now empty, during that period of time, you're in this relative refractory period as that tank begins filling. You could hit it and flush it again, but it would be different. In reality, the best way to make this analogy work would be if during that time the tank is filling, you had to push on the handle harder to make it flush normally. That's the perfect analogy for this one. We know that's not the case with a toilet, but gives you this idea. Once your toilet has completed its flush, you can get it to try to flush again, but it's during that relative refractory period until the tank fills, so you get a different flush in the toilet. Again, what this means for us in terms of action potentials is it means you need a stronger stimulus to send action potential, or quicker action potential after the first one. So you're going to need a bigger stimulus. It's going to have to be able to move at more millivolts than it normally would. So instead of only doing 20 millivolts, it's going to be able to have to move at 25 or 30 millivolts if you're in that relative refractory period in that undershoot. So like I said, if you're right here, if this was negative, let's say this is negative 70, let's say threshold is up here at negative 50. If this is negative 80, it's 30 millivolts to reach threshold when you're in the undershoot, where from here it's only 20 millivolts to reach threshold. So in this red box here, that is that absolute refractory period. When you're in the process of depolarizing and repolarizing, you can't send another action potential. It's only after you've repolarized, and again, until it gets back to that resting potential, you're going to need a bigger stimulus to send a more frequent action potential. So we've talked about these action potentials now, and I've kind of said they are all or none. Either you send an action potential or you don't. It either reaches threshold, it sends an action potential, or it doesn't. what this also means when we say it's all or none, it either sends the same action potential or it doesn't send one. It doesn't send a half strength one. It doesn't send a double strength one. So yeah, the bottom question there, will one action potential ever be bigger than another? The answer to that is no. None of them will be bigger than the other. They'll always be the same. So if there's a bigger stimulus or a smaller stimulus, how do we convey that information, I guess, is the next question that everybody needs to think about. So when you are now at this one, okay, we had a much bigger stimulus. It really passed threshold. How can we send that information with an action potential? And that's what we need to think about here as we start talking about a little bit more of this. And that's getting into how do we interpret these ones. So how do we deal with an action potential if it's all or nothing and it always sends the same size? How do we convey any information in an action potential? And the answer to this one is a little bit that we don't convey it in a single action potential. A bigger stimulus, so a larger stimulus, is going to mean that you've reached that threshold by a bigger value. So we had a bigger change in that membrane potential. So instead of just reaching threshold, we might be 30 or 40 millivolts change in that membrane potential. What that means is as soon as that gets into that relative refractory period, you could send a message again. So how we send information about the size of stimulus and an action potential that is all or none, we don't send it in a single action potential, but a larger stimulus means we can reach threshold value again quicker. So back during that undershoot. So how we send information about a bigger stimulus is we send more frequent action potentials. So if we have more frequent action potentials, it means the stimulus was larger because we were able to send an action potential as soon as it got repolarized, even though it was, let's say, 30 millivolts away instead of 20. We had enough of a stimulus that it was able to reach threshold immediately once the membrane was repolarized. So frequency of action potentials is how we send information about the size of the stimulus. And if we look at the tail end of this, it's also going to convey that to the next cell, because if you're sending more action potentials, you're going to release more neurotransmitter versus sending less. So the stimulus to the next cell is going to be larger. So that's how we send information in these ones. It's the frequency a lot of times, not the size of the action potential, because that's always going to be the same. It's the frequency of action potentials that are going to convey information about stimulus size or how big that initiating stimulus was. And again, that information is going to get down to the end of the synaptic end bulb. It's going to cause a depolarization. It causes these vesicles to merge here and release that neurotransmitter into the synapse. At this point, we're converting our electrical signal back to a chemical signal, which will allow it to talk to the next cell. And again, this is due to voltage-gated calcium channels at the end of this here. We have extracellular calcium. When this gets down here, this extracellular calcium floods in here when we open up these voltage-gated calcium gates. These merge with the whole cell signaling pathway to cause these synaptic vesicles to merge with the membrane on these little docking proteins and dump their neurotransmitters out. They're going to diffuse across the synapse and they're going to bind to receptors on the other side and initiate some type of response. One of the things that is very interesting with nervous system, when we talked about the muscles and this idea of binding to a ligand-gated channel, the only thing we talked about were ones that were going to be stimulatory, so bring it closer to threshold. Again, with a muscle, when you've decided to contract a muscle, you've done the processing already. We're just trying to get that to happen. When we're doing processing within the nervous system, we're going to have different stimuluses, and again, you're going to have multiple synapses on any particular neuron. And again, we've said that these neurons need to be able to process information, do this parallel processing. What this kind of means is any particular neuron, it may have multiple synapses. And at these different synapses, there may be different neurotransmitters. The one thing we're going to see with neurotransmitters is not all neurotransmitters bring about a stimulatory effect. They're not always going to get the cell closer to sending an impulse. Some of these neurotransmitters will bring about this graded depolarization. They will cause positive ions to travel into the cell, make that cell more positive. That is going to be a stimulatory type of effect of a neurotransmitter. But there is also some neurotransmitters that cause negative ion channels or anion channels to open up. And what that's going to mean is you let negative ions into the cell. If it's normally negative 70 in the cell, and we let more negative ions into the cell, so let's say chlorine ions into the cell that are negative, instead of being at negative 70, it might get down to negative 80. at that point, we're further away from threshold. So further away from those voltage-gated channels being triggered to bring about an action potential down the axon. This is what we refer to as a graded hyperpolarization, and this would have an inhibitory effect on this neuron sending a message. So what we're going to see, the processing that takes place here, is it's going to be a combination of all the synapses that this, on a dendrite, let's say, all the synapses that this neuron has. There might be some that are stimulatory, some that are inhibitory. If we stimulate both these ones, it's going to be what happens to the electrical in here. Was there more stimulatory responses than inhibitory ones? If it gets to threshold at the action potential, it sends a message. If it doesn't, it doesn't. We're going to see that there is some neurons that have 200,000 synapses. Again, the whole decision-making process is, does it reach threshold or not? If within these 200,000 synapses, if we get enough stimulatory messages, it may send a message from that neuron. Again, and that's not even getting into what might the message be that that neuron sends. It might be a stimulatory message to the next neuron. It might be an inhibitory message. We don't really know that. But again, the hyperpolarization, if the inside of that becomes more negative, that could mean we open up more potassium channels and let more out, or we open up more chlorine ion channels and let more in there. Either way, if we get the cell more negative, it's further away from threshold. That is that idea of hyperpolarization. It is going to be inhibitory. And again, this neuron that is receiving signals, it may be having multiple synapses with multiple neurotransmitters all talking to it at once. And we may be getting graded excitatory and inhibitory messages all at once into this neuron. It's just, again, whether or not does it reach the threshold value of the voltage-gated channels at the axon hillock. So we can talk about, actually, these ideas of, so this is an excitatory postsynaptic potential. So excitatory postsynaptic potentials, or EPSPs, these are going to be neurotransmitter releases that are going to bind receptors that are going to open up positive ion channels that let more sodium either get into the cell than the amount of potassium that's getting out. This is going to get the inside of the cell slightly more positive, closer to that threshold value. If it reaches threshold value, we might get a signal. So that would be an EPSP. The degree of change here, again, depends on how much neurotransmitter, how long. If we get enough excitatory neurotransmitter releases, we can reach this threshold value. It gets closer, or get closer to the threshold value. If it gets to the threshold value, that neuron will send an action potential. So again, an EPSP would be something that gets it closer to the threshold value. So we let some sodium ions into the cell. Again, if we get enough of these, it may reach this number here. Then we bring about an action potential that sends a message down that axon. We can also have inhibitory postsynaptic potentials. So inhibitory postsynaptic potential, IPSP. In these cases here, these might bind to potassium channels or chlorine channels, either let more potassium out of the cell, which is going to make the inside of the cell more negative, or let chlorine ions into the cell, which would also make the inside of the cell more negative. If the cell gets more negative, that is going to move us further away from that threshold value. And again, these are all local. They are spreading out, just like we saw in other ones. But again, if we get an inhibitory message, you can see it gets further away from the threshold value, which means it's going to take a bigger stimulus to reach that threshold value. And again, it's kind of the combination of these inhibitory messages and excitatory messages that are going to determine whether or not this cell is going to send a message down its axon. Again, both of them being released at once, maybe more frequent or less frequent. You can have them all going on at once, but again, the processing takes place in that, does the overall electrical change in the cell get to the threshold value at the axon hillock and allow that message to be sent, or does it not? So, and again, you could have something like this, where you have this multipolar neuron with lots of these different synapses here. And again, does the combination of all these together reach the threshold value down here or not? Another thing I just want to talk about that is kind of interesting is also this idea of, could one of these synapses be having a bigger effect than another? And you might kind of go, well, no, they're all kind of combining. What's kind of interesting on this one is these are local effects, right? So let's say this stimulatory one out here. If it causes an opening here, you're going to get these, let's say it was an EPSP, so an excitatory message here. It's going to have sodium ions flooding in, and it's going to spread out from here. As they spread out, they're going to get weaker. But on this dendrite, it's quite far away from the axon hillock. It's going to take a bigger stimulus here to get a big enough change to cause it to reach threshold here. Now, if we're looking at the one that's right next to it, could a change in the membrane potential here have a bigger effect? The answer is yes. If you have a synapse that is much closer to the axon hillock, it's not going to take as big a signal. So again, where these synapses with this particular neuron can also have an effect on how big of a role they play in the processing of this ones. There might be certain synapses that neuron is gonna play a bigger role in, does it send an action potential or not. So ones that are closer, either excitatory or inhibitory, that are closer to the axon hillock and those voltage gated channels are gonna have a bigger effect in terms of, does this reach threshold or not. It's not gonna take as big a signal from these ones to either generate an action potential from the excitatory ones or inhibit one from the inhibitory ones if they're closer to the axon hillock. Again, just something to think about. And again, this is talking about these multipolar neurons. Again, with how these are laid out on, let's say, unipolar neurons, all that processing, when you remember that one, it had a dendrite here, the cell body was off in a little T, something like this. All that processing is taking place up in the dendrites on a unipolar neuron. On a bipolar neuron, it's taking some processing here, gets to the cell body, and then, again, does it reach threshold of the cell body to send it down the axon of that. So they're a little bit different on some of these other ones here. I'm not going to ask questions about how does an action potential differ on a unipolar versus a bipolar versus a multipolar. I just want you to understand that the processing in those based on how they're built is a little bit different. But again, understanding that certain synapses may play a more key role in whether or not it reaches the threshold or not, that's, again, not something I'm probably going to ask you on a question, but it's something to think about when you're talking about how does a nerve do this processing, how does all this go in there. And then again, to me, this is really neat when you start talking about the billions and billions of neurons that are in your brain. This is how you think. This is how you have personality. This is how you decide whether or not to do a muscle movement or not, how you decide whether or not to blink. It is all this processing. It's all this parallel processing where you have multiple inputs and this one cell with all these inputs determining whether or not it sends an output. And then we're doing that a billions of times over with all these trillions and trillions of connections in between these cells. So again, very neat stuff. Stuff that we probably don't understand as well as we do a lot of other parts of the body. So when we talk about this combination of all these EPSPs and IPSPs, we talk about the idea of summation. This is kind of this idea that all these electrical changes are going to add together. Do they reach that threshold value, that negative 55? That means we have to get a 15 millivolt change from resting potential. If threshold is reached, those channels open. If it doesn't, they don't. And the main thing is, excitatory messages will get us closer to that threshold value ones. Inhibitory ones will get us further away, and if we're receiving both inhibitory and excitatory messages, the inhibitory ones are going to kind of block the effects of the excitatory ones. And again, it's just, do we get enough to reach a change here? So it's kind of, if we went back to our analogy of a house, if you had doors and windows, certain ones that were hot air and certain ones that were cold air, if you opened up a certain door that was a warm air door and a door that was a cold air door, did the temperature in the house change enough to cause your thermostat to drop enough to kick it on? The furnace? Yes or no? That's the idea of this summation, this adding up of all these effects. And what we're going to see is you could add these up over time, so temporal summation. So the same synapse kept firing a message, or with the house analogy, if you went to the door and you kept opening and closing the door, you're going to get burst of cold air, burst of cold air, burst of cold air. If we add that up over time, it may get the house cold enough that the furnace kicks on. The other thing you could do is have multiple excitatory messages all at once from different synapses, or if we go back to the house analogy, you opened up multiple doors and multiple windows, and all that cold air coming in at once from all these different openings caused you to reach the threshold value on your thermostat and cause the furnace to kick on. That's the idea of spatial summation. So again, this to me is one of the tougher videos that we're going to be doing on here. This is that idea of processing and how these determine what an action potential means. Again, if there's questions on this stuff, always you can contact me, other stuff like that, and we can figure it out. So I'll talk to you next time.